Hey guys, welcome back. We're going to continue on with photosynthesis today. Pardon me. We left off last time, we spent a lot of time talking about the light-dependent reactions of photosynthesis, and hopefully you'll recall two products that we were really spending a lot of time on, and those two products were NADPH and ATP. So today we're going to transition into our light-independent reactions. Now, right away, I don't want you to be fooled by this name. Light independent means it kind of gives you this impression that it doesn't depend on light, but bear in mind that it does depend upon the products of the light dependent reactions. So even though I don't directly need photons to start this part of photosynthesis, I do require those products of the ATP and the NADPH that we wouldn't have if it weren't for photons. So hopefully I didn't just make that more confusing for you, but that's light-independent reactions. So let's jump right into it here. Energy flow in our light-dependent reactions is an example of how organisms harvest energy from their environment. Okay, no duh. That's what we've been talking about, right? Plants make sugars from the sun. That's harnessing energy. That's what that is. And so we're producing NADPH, and this is light dependent still, and we know that. And we're going to get that into the light independent reaction, which is what we're concerned with right now for this set of the lecture. So our light independent reactions are cyclic. A lot of what we learned about last time was, I guess, non-cyclic or linear, where it had a start and it had an end. This one, we're going to call it a cycle, the Calvin-Benson cycle, and it's sometimes just shortened up to Calvin cycle, is going to be cyclic because some of the products, if you will, of the cycle are going to serve as the reactants to start the next turn of that cycle. And so it is not linear, it is cyclic, and this is the synthesis part of photosynthesis, because up until now we've made a lot of energy, and now what we're going to do is take that energy and we are going to synthesize glucose. And I can't overemphasize the importance of glucose to plants and to ecosystems as a whole, because really that's where we take energy from the sun, which is not so useful, and turn it into something that's useful for pretty much all of our animals and everything that depends upon the plants, and the algae for that matter. So these are enzyme-mediated reactions, which means there's going to be enzymes that we're using that build sugars in the chloroplast, and we are in the stroma still. We are in the fluid inside the chloroplasts, not on the thylakoid discs. So hopefully that's clear. So here we go. We call this carbon fixation because we fix carbon into a usable form. And so we extract carbon from inorganic sources like the atmosphere. So CO2, we can't use that. That's not that useful for really anything from an energy point of view. And then we get them to an organic molecule, which is glucose. So 6 carbons long, and that holds a lot of energy and is digestible by most animals and bacteria and fungi, et cetera, et cetera. And so it's very, very important that we accomplish that. We use the bond energy of our molecules that we formed in those light-dependent reactions we talked about last time. ATP, there's a lot of energy there. NADPH, there's a lot of energy there. And so we need these guys to get carbon dioxide turned into glucose. And that process from carbon dioxide into glucose is the Calvin-Benson cycle. So let's jump right into it. The Calvin-Benson cycle starts with the enzyme called Rubisco, sometimes called Rubisco BP. And food for thought, this is most certainly the most abundant protein that we know of on our planet. This is really, it's that important. So the RUBP is what we're going to connect CO2 with, and that's why he's so abundant. And so the enzyme Rubisco attaches carbon dioxide to RUBP, and when it does that, it forms these 3 carbon molecules we call PGA. And so PGAs become phosphorylized from ATP, and they also get hydrogen and electrons, some more energy, from NADPH. So there's both of our products from the light dependent now being used just right away, really in the second step of the Calvin-Benson cycle. And so once we attach these hydrogens, we turn PGA into what's called phosphoglyceraldehyde. so PGAL, and those guys are 3 carbons long, and we can combine 2 of these to form a 6-carbon sugar, and that 6-carbon sugar, you guessed it, is going to be glucose. And then at the end of this, the cool thing is this enzyme we started with, Rubisco, is regenerated. So the guy that we started with, let's see here, pen, this fellow right here, we get at the end, and he can go right back in and run this cycle again, and right back in and run this cycle again. Now that's the nice news. Here's the not so nice news. This cycle that we're describing, 1, it's a little wordy and tough to kind of memorize. 2, it is very expensive. It costs a lot of ATP. It costs a lot of NADPH to make glucose, but it's worth the cost because glucose is energy storage, and glucose is the building block for cellulose, which is cell walls, and glucose it's absolutely essential. So it's worth the cost, but do know that the cycle is expensive. And so here it is, it's a step-by-step breakdown. We start with our RUBP, and we combine carbon dioxide with it, and we make PGA. And you'll see that we're making 6 RUBP into 12 PGA, which kind of makes sense because this guy here is 1, 2, 3, 4, 5 carbons. We bring this in, now we have 6. Okay, so we've got 6 carbons, and we break those down into 3 PGA. So our PGA, which is 3 carbons long, we give it a phosphate group from ATP, and look at how much it costs. It costs us 12 ATP. That's a lot. It costs us 12 NADPH. That's a lot. But when it's all done, we've converted PGA into PGAL. Now, that's step 2. This is 12 of these 3-carbon pGal molecules. So 2 of these 3-carbon pGal molecules will combine to form 1, 2, 3. There's one of them. 1, 2, 3. There's the other pGal. A 6-carbon glucose, who is our simple monomer building block unit for really all sugars. And it can go on to turn glucose into sucrose, and we can transport that around through the plant's phloem tissues as sucrose, and we can get them down to the roots, and we can link them up and build this big starchy carrot or potato, or we can get him to cell walls, and we can turn him into cellulose. There's a lot we can do with him. He's very, very important. So this is our product. This is what we're interested in. So the cool thing is we only took 2 of these pGals to make glucose, and we have 10 left over. And so the 10 that are left over go back into the cycle. So they're going to give up that inorganic phosphate, a few of them. Bear in mind, we started with 12. Okay, we used a lot to get to this process, but we've got 4 we can give up. And then we're going to bring in some more ATP because we need more energy. You notice we've already started with 12, now we got 6 more. We're taking a lot of ATP really, and we're going to regenerate. By using this ATP, we are going to regenerate that same RUBP that we started with. And then around and around it goes, and that's why it's called the Calvin-Benson cycle. So this diagram, just so you know, they tried to keep their thylakoid discs on the outside of the chloroplast. It's not necessarily terribly realistic, but they wanted to provide enough space to show you that this was in the stroma, not on the thylakoids. So do bear that in mind. All right, and so here it is, another cartoon all together, because the last one they blocked out RUBP at the end. So we start with RUBP, carbon dioxide, PGA, ATP, NADPH, pGal, sugar, leftover pGal, more ATP, back to RUBP. So I know that's a lot, and it is a lot to take in, but it is important to know what our inputs and outputs would be. You know, so our inputs are certainly going to be carbon dioxide, RUBP, ATP, NADPH. And then our outputs are going to primarily be glucose and RUBP. And so it is very important to understand that. I've got a little video for you, and hopefully that helps clarify things. Let's take a look at this.
The light-independent reactions make sugars by way of a cyclic pathway called the Calvin-Benson cycle. The cycle begins when Rubisco attaches a carbon from carbon dioxide to ribulose bisphosphate. The molecule it forms splits into two molecules of PGA. Each PGA gets a phosphate group from ATP and electrons from NADPH. The resulting intermediate, PGAL, is primed for reaction. After 6 molecules of carbon dioxide have been fixed, 12 PGAL form. 2 of the PGAL molecules combine to form the 6-carbon sugar glucose. The glucose will enter other reactions that form carbohydrates. The remaining 10 PGAL get phosphate groups from ATP. This primes them for the reactions that regenerate RUBP, thus completing the cycle.
All right, so very similar little cartoon to what we just saw in the PowerPoint from our book. We are still seeing the inputs of ATP and NADPH, along with our outputs of glucose and our RUBP. So I know we're not having a particularly long video this time, but it's a lot to try to learn this cycle, and I do want you guys to understand this cycle, what goes in, what comes out, what are the intermediates. So we're actually going to call it there. Next time we'll come back and we'll talk about some deviations to this, C4 plants and CAM plants and some stuff you might have learned a little bit in Gen Bio or Botany. But for right now, I think that's enough to kind of just sit back and digest for a little bit, organize your notes for it. So thanks for your time.